Hi, everyone. Super happy to be here. Thank you for the invitation. This is my first meeting. And today I'm going to talk about, indeed, how we're using brain organoids for understanding STXBP1. I hope that our preliminary results will not cause that much havoc, so bear with me. I always start with this slide, which is the very good driver. Finding drugs for neurobiological disorders is really, really hard, especially neurodevelopmental disorders. And this slide is not a huge encouragement for the biotech companies. It just suggests that the more money you put into finding a neuro-related drug, the more money you're likely going to lose. So there's several reasons for that. One reason is that we don't have enough of good human models. And this schematic shows our approach, which is using human brain organoids. I'm not going to introduce them that deeply, but the idea is that you start from induced pluripotent stem cells that you can take from healthy individuals or from patients, and you reprogram them, and then you differentiate them in 3 dimensions to generate a 3-dimensional growing developmentally related human brain culture or cell culture. And organoids contain the various cell types of the brain. So as far as we know, they are the best, most physiological human model to model early parts, at least, of development. And in this case, I showcase an example of how we're creating human brain organoids. Another added feature or advantage that they have is that they enable creating specific brain regions by patterning the human pluripotent stem cells to specific fates, specific regions. So in this case, it shows how a human cortical organoid is generated. You start from stem cells, then you kind of round them to little spheres, and then you start a differentiation process. At the end, you get a human cortical organoid. If you look inside, you can see the relevant developing progenitors, later also neurons, and if you put an electrode inside, you might even find some electrical activity. Okay, this shows well. So when I approached this, I had a few questions that I was trying to answer. How to use organoids in the best way to approach STXBP1. So there are several considerations. First, why in the first place use human and organoids? So what I like about them is that they have, first, they're human, and they're based on human genetics, but they're also highly scalable and relatively reproducible compared to other methods, and they're kind of reproducible across labs, at least. Then which brain region to choose? What are we going to study? I like studying the cortex, but in the context of STXBP1, it seems to be highly relevant because of the cognitive-related needs and the seizures. Then another problem was which mutation am I going to check? And there's a lot of assumptions coming from the literature that basically, after reading a lot of paper, I ended up really, really confused, not knowing what's the best mutation to study. And eventually, we ended up studying one mutation instead of studying multiple mutations, because it allows some depth and maybe deeper understanding of the pathogenesis. And it's going to be R292C. And then another question is which cell type? Cortex has different cell types coming from different regions, and everything is in the mix. So that's another quandary. And when during maturation, that's another question it already has raised before in the previous session. So luckily for us, through Rafa's moonshot and Gad Vatin's lab from Ben-Gurion University, they generated an R292C line from a patient who carries this specific mutation and from a familial control. Some of you guys know them, but I'm not going to expose them here. And a PhD student in my lab, Lilach Mekonet, got a hold of the lines. She started growing them, characterizing them, seeing that the mutation is indeed what we think it is, and And basically from that point on, she took them and started creating cortical organoids. And one of the first data sets that we created was around day 80. We harvested the organoids and took them for some bulk RNA sequencing and looked for what kind of regions are we making. So a secret in our field is that you start with iPSCs, but different lines behave differently. So one conclusion was that most of the signature, sorry, maybe I should go there. Most of the signature directs to a pallium fate. So the organoids seem to be from both lines, the control and the mutant, patterning to a dorsal pallium fate, although it's not perfect. Then we subjected them to immunostainings to see kind of a sanity check to see what we're seeing inside. So around day 80, we immunostained them, and we stained them for a neuronal marker, something like MAP2, and also for, NEO-N, and for STXBP1. So one result that was interesting was indeed we saw in this mutant a reduced levels, at least by immunofluorescence, not the best method. We saw reduced STXBP1 levels. At the same time, we also noted that the cell bodies in the mutant appear to be larger around this time point. This is another thing. We quantified the surface area of the neurons. So the neurons in the mutant were both larger and they were less dense. So that suggested maybe there's something going on with the morphology of the cells, although we don't have a definitive answer yet. Good. So the next step was to take the organoids, and around day 80, we infect them with an AAV GComp, and then we image them. The movie should play, but it's not, or maybe it's just hard to see. Okay, good. So to our surprise, I wasn't that surprised, but I was a bit surprised. What you see on the left are the control HCOs, and there's very little activity. There's a few cells that are flickering around. Most of their activity is completely asynchronous, and we're seeing this consistently with this specific protocol across many, many lines. So in that sense, the control line behaved just the way that we expected it to behave, while the STXBP1 mutant line behaved totally differently. It was more cells were active, and when they were active, they were firing those kind of little bursts of activity, and the bursts of activity were kind of intermittent, going on and off, and involved a lot of the cells. The neurons were completely synchronized. We quantified this, and this kind of made sense in the sense that we're seeing more synchrony and more activity, but we didn't really know what's going on. So a few potential explanations to our phenotype in this R292C HCO is first, for everyone here, seeing this kind of activity in vitro. It means it's not epilepsy and it's not even seizures. At least in electrophysiological terms, this is not how we define them. But there is increased activity, and there is increased synchrony, and we also see those kind of bursts. So there's all kind of, I'm an electrophysiologist by training, so there's all kind of considerations to this. It could be an intrinsic effect, it could be a synaptic effect, it could also be a maturation effect. And it could also be, unfortunately for me, it could also be a cellular composition effect. So when you work with organoids, when you pattern them like that, many times you end up with slightly different cellular compositions. It could be another explanation. So one way to approach this was to use a little bit of pharmacology. We wanted to see if the controls can, in principle, even at this specific time points of around day 95 in culture, behave like the mutants. So what we did was subjected them to 5 minutes? Okay, not that much time. We subjected them to a pharmacological agent called 4AP. It increases both intrinsic activity, so how much the neuron will fire for a given input, and how much, and also synaptic excitability, how much it will communicate its activity to the network. And the controls, at least at this time point, did start to fire more, but they didn't really synchronize. They started showing a little bit of bursting. The mutants, on the other hand, once we subjected them to 4AP, they went completely crazy. They fired an endless amount of action potential, so synaptic activity. And they remained highly synchronized. We then tested again 20 days later, so this is day 115. We again tested the controls with 4AP, but now all of a sudden, the controls started to behave a little bit like the mutant. While when we treated the mutants, they again went to this berserk mode where they just fire incessantly. So this is our major finding so far. And I'll just give a short summary about this, and maybe I give a few more sentences about what we're trying to do now. So what we did is we established those two lines using the HCO protocol, and we found that they map okay to the dorsal pallium. Sorry about that. And we find morphological and functional phenotypes that will allow us to screen putative rescue approaches. And one potential explanation for our effect, I think you can come and ask me about this, is enhanced or more rapid synaptic and intrinsic maturation. Okay, if I just have a few more minutes, if I was able to convince you why organoids are so valuable, now I'll try to convince you that they're not so valuable. Because everyone knows that they have this problem with maturation. And our claim to fame in the lab is to take organoids, and when they're 30 days old, we transplant them into newborn rats. And in the newborn rats, they create these huge grafts, and inside each one of those grafts, you get beautiful human pyramidal neurons that are, as far as we know, the best replica of a human pyramidal neuron around 8 or 9 months from embryogenesis. And they also can answer, those kind of neurons are more relevant for the time windows that we were discussing before, because they just look way more like what we would ideally like to treat one day. And in the past, we used them to identify phenotypes in a disease called Timothy syndrome. And later, we also used these grafts to test an ASO, antisense oligonucleotide medicine in a living animal on the human cells successfully. So they are super promising. And this is something we're doing now. We're transplanting organoids of both excitatory and inhibitory lineages. And we have them already in the animal room. Next week, they will be patched. And ongoing work will kind of involve both the dorsal organoids and dorsal and ventrally patterned organoids, transplanted and non-transplanted. So our ongoing work is validation of the HCO phenotypes in another new pair of iPSC lines. We're testing the efficacy of NMN and an ASO on our organoids, and we're trying to identify more mature defects in R292C HCO neurons, and we will also look at dorsal and ventral integration of regionally patterned organoids. And lastly, we maybe have this kind of a hope that we will see maybe seizure-related activity in our transplanted organoids because of their maturation. And finally, this will all allow us for identifying or maybe testing new therapies, which is, we're excited about this. And I'm excited to talk to people who have those therapies come to me. That's it. Thank you. Thank you for listening, and thank you for the student who led the results. So, I don't know if I have time for questions.
Hello. Thank you for this really interesting talk and like an advance on the brain organoid strategy. Once you have the organoid in the rat, then how do you evaluate it? What kind of endpoints can you use after you've made that, that method?
Thanks, great question. We made all the possible endpoints possible. So you can get tissue, you do immunostainings, you can record activity in vivo, you can use optogenetics. We went all the way to even use organoids to engage behavior in the animal, and recently induce a seizure in the animal by triggering a seizure in the organoid. So I don't know if we can do everything. We're still relatively small lab, but
that was a great talk. Thanks so much. Did you look at the intrinsic excitability of these neurons?
I knew that you will ask that, but it seems that, so we didn't patch the in vitro organoids because I assumed that I will see a small phenotype. By the time I found that the phenotype was huge, we kind of discarded the batch and we will do it. We did have some patching going on from transplanted organoids, but they didn't have a good control. The little ones, the few that we did show increased excitability. Like, yeah, I cannot say it yet. That was the tendency.
Looking forward to that. Thanks.
So when you do the transplantation, did you see the organoid connected with the host brain, like
yes.
a vesicle and the synapses?
Yes, that's totally. I didn't discuss this at all, but indeed it receives connections from the host and sends functional connections or functional outputs into the host brain. Okay, thank you.